Hello students, welcome to 7th lecture of biomolecules. In previous lecture, at last we discussed about primary structure of protein, and in this point a sentence was mentioned: each polypeptide in protein has amino acid linked with each other in a specific sequence, and it is this sequence of amino acid that is said to be primary structure of protein. So what it means, let us talk about it first. Let's say our protein is made up of three different amino acids: glycine, alanine, and valine. So their initials are G, A and V respectively. So suppose in our amino acid our sequence is G, then we have A, and then we have V. So this sequence of amino acid will repeat in entire structure of polypeptide or protein. So same type of sequence will be observed in whole protein. Suppose if we use same three amino acids in different sequence, that means as for example like this G, V, A, and same type of repetition will be observed G, V, A in entire protein, then this protein is completely different from the first protein. So that means not only the amino acids but their sequence are also important for protein. If you change the sequence, then you will get a new type of protein. Now the secondary structure of protein refers to the shape in which long polypeptide chains can exist. They are found to exist in two different types of structures, that is alpha helix and beta plated sheet structure. This structure arises due to the regular folding of the backbone of polypeptide chain due to the hydrogen bonding between C double bond O and NH group of the peptide bonds. Alpha helix is one of the most common way in which polypeptide chain forms all possible hydrogen bonds by twisting itself in a right-handed screw or helix with the NH group of each amino acid residue hydrogen bonded to the CO of the adjacent turn of the helix as shown in figure 4.1. So as we can see in this structure, and as we know that we will get multiple different type of amide bonds or peptide bonds when multiple different amino acids join together. So NH2 group and COOH group, when joined together, it will release water molecule and you will get CONH2 bond. So this CONH2 bond contains hydrogen attached with nitrogen and carbon attached with double bonded oxygen. So there is a possibility for the hydrogen bonding between hydrogen, that means NH group, and CO group oxygen. So they will attract each other like this. Suppose we have CO bond of the one and NH group of the other peptide, then they will, if they are comparatively close enough, then they can form hydrogen bonding, because our requirement is hydrogen should be attached with highest electronegative elements, fluorine or oxygen or nitrogen. And if we place one of these three closer to that hydrogen, then hydrogen bond can be possible. So suppose you have this type of primary structure of polypeptide or protein, then you can see that we have multiple hydrogens attached with nitrogens and multiple oxygens available. So when we twist this structure like this, then there is a possibility hydrogen attached with nitrogen and oxygen attached with carbon can form hydrogen bond between them, and this is the reason why this type of coiled structure is obtained. So this type of helix structure will be obtained because of self attraction. That means we can say that this is because of intramolecular hydrogen bondings. We have a single structure of protein and it is coiled due to the hydrogen bonding proceeded between their different parts. And as we can see here, oxygen is attracting hydrogen attached with nitrogen. That means this is a type of hydrogen bonding. So all of the dotted lines mentioned in this structure are hydrogen bondings, and because of this hydrogen bonding you will get this type of coiled structure of our amino acid, polypeptide or protein. Now in second type of secondary structure, that means beta plated sheet structure, all the peptide chains are stretched out to nearly maximum extension and then laid side by side, which are held together by intermolecular hydrogen bonding. That means in previous case helix structure we have intramolecular attraction, and in this case we are placing two same type of chains closer together and then they will attract each other because of hydrogen bondings. So it is intermolecular hydrogen bonding, and the structure resembles the plated folds of drapery, and therefore it is known as beta plated sheet. And this is the example of drapery, that means folds in clothes which are properly arranged. So assume you have two different type of these type of clothes folded equally and then placed on top of each other. So this is the example of beta plated sheet structure. Here we have a rough representation of beta plated sheet. Suppose this is our first protein or polypeptide, and this is how it is zigzagly arranged. And now as you can see, in certain points we have oxygens and certain point has hydrogens coming out of the structure, and here we placed second same type of structure. Okay, and as we can see, in certain point hydrogen is coming out, certain point oxygen is coming out, and when oxygen of first and hydrogen of second, or vice versa, hydrogen of first and oxygen of second is poking out, and if they are comparatively close, then hydrogen bond will arise. Okay, so this type of multiple plates will be present close to each other, and that's why this type of beta plated sheet structure will be obtained. Then the tertiary structure of protein represents overall folding of the polypeptide chains, that is further folding of the secondary structure. It gives rise to two major molecular shapes, that is fibrous and globular protein. We already discussed about these two shapes. The main force which stabilizes secondary and tertiary structure of proteins are hydrogen bond disulfide linkage, van der Waals, and electrostatic force of attraction. For sulfide type of disulfide type of linkage, we need some specific type of amino acids present in proteins, because we need sulfur to form disulfide bonds, while the remaining hydrogen, van der Waals, and electrostatic force will be present in all of the proteins, because all of them contains certain portion which can produce hydrogen bondings, and van der Waals force of attraction is applicable for each and every type of particles. Now question may arise, why van der Waals force of attraction is mentioned, because it is very weak attraction. But if we have multiple different type of van der Waals sites, that means locations where these type of forces are produced, then overall they will give us comparatively very strong effect. That means, let us say strength of single van der Waals bond is, let us say, 2 kilojoules. Normally hydrogen bonds have 40 kilojoules of energy. So one simple hydrogen bonding is comparatively far more stronger than normal van der Waals bond. But suppose you have 100 van der Waals bonds or locations or attractions, then 1 is equal to 2, then 100 is equal to 200 kilojoules of energy. To remove 100 van der Waals locations or attractions, we need to apply 200 kilojoules of energy. And as we know, proteins are very large molecules, so obviously they will produce multiple different type of van der Waals attraction, and that's why it will stabilize it. Now next is a quaternary structure of protein. Some of the proteins are composed of two or more polypeptide chains referred as subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. That means, suppose you have tertiary structure of protein, that means very coiled structure of protein, and when we mix two different type of proteins or more than two different type of tertiary proteins with each other in specific order or arrangement, then you will get quaternary structure. That means, as we can see in this case, we have all of the representations of different proteins. Here we have beads type of arrangement in first case, and where all the beads represents amino acid, and this is primary structure. When this type of long chain of peptide, polypeptide or proteins gives these two type of structure, that means plated sheet type of structure or helix structure, then it is secondary type of protein. When these type of plates or helix even further coiled like this, then it is tertiary structure of protein. And this black line actually represents, let us say this is helix structure of protein. Okay, so this is helix structure of protein even further coiled to give us tertiary type of structure. Okay, so this type of arrangement of further folding of secondary type of structure gives tertiary type of structure. Now assume this tertiary type of structure as a single unit for quaternary structure. That means we have to arrange at least two these type of tertiary structure to get quaternary structure, as we can see. Here we have one subunit represented as light blue color and second subunit as dark blue color. So these two different subunits are arranged closer together, and some portion will be mixed together to give us a quaternary protein. That means at least you need two different tertiary proteins to create quaternary protein, and you can have more than two. And here we have representation of all the four type of proteins. So in this case, first structure is primary protein, where these colored dots or beads represents amino acid units. When these amino acid units coiled together like this, as we can see in this second structure, which is helix structure, then it is secondary structure. Then this helix structure further folds like this, you will get tertiary type of structure. And when we have multiple tertiary type of structures coiled in between or together, then you will get this type of quaternary structure. In this case, we have two different type of tertiary structures. First is just like this tertiary structure we have here. Okay, so this is our first tertiary structure, and then we have secondary structure starting from here and coiled with first tertiary structure. And in this second representation, same type of structures are mentioned with some complex geometries. So this is amino acid units which represents primary structure. When this primary structure coiled like this, you will get secondary. When secondary structure even further coiled, that means if you can see, this is yellow strand of protein is actually secondary structure, and this secondary structure is even further coiled, so you will get tertiary structure. And when you have more than one these type of tertiary structure coiled together, it is quaternary structures. Our last point for protein is denaturation of protein. Protein found in biological system with unique three-dimensional structure and biological activity is called native proteins. When protein in its native form is subjected to physical change like change in temperature or chemical change like change in pH, the hydrogen bonds are disturbed, or any type of intermolecular attractions will be disturbed. Due to this, globules unfold and helix get uncoiled and protein loses its biological activity. So keep in mind, in our body many different type of proteins do exist, and all of them are functioning due to their specific shapes and coiled structures. If they loses their shape, then they will lose their biological activities, and this is called denaturation of proteins. And denaturation can destroy secondary, tertiary and quaternary structures, but primary structures will remain, because primary structure are not because of intermolecular attraction, it is because of permanent bondings between the atoms present in amino acids. Whereas secondary, tertiary or quaternary structures are because of intermolecular attractions or intramolecular attractions. So change in chemical or physical state of the protein can disturb inter or intramolecular forces, but it cannot disturb permanent bondings. That's why primary structure will remain intact. The coagulation of egg white in boiling is common example of denaturation. Other example is curdling of milk, which is caused due to the formation of lactic acid by bacteria present in milk. So our second biomolecule protein is completed. If you find any difficulty regarding to the protein, feel free to ask, and in next lecture we are going to move on enzymes.